Okay, so I told you what cerebrum did, right?
Cerebellum.
Processing of senses, be it somatic senses or sensory, like special senses. Somatic senses would be like touch, heat, that type of thing, pressure. And special senses would be like sight, hearing, smell, taste, that type of thing. Well, cerebellum is kind of motor function. So it helps to coordinate muscular activity. It helps to maintain posture and equilibrium, and coordination of movement. Okay, so in cerebellum, definitely a difference between its appearance and cerebrum, right? This has a weird stain on it that made the white matter black. That is the white matter all through here. The thing that looks like tree branches, kind of. So there's the trunk of the tree coming out through the branches, out there, and out through here. And then you have 3 layers of gray matter. Gray matter, gray matter, and gray matter. Come from up this way. Okay, so this is your first layer of gray matter. Then you have this really thick, dark line of cells through here. Those are a special type of neuron called Purkinje cells. That is characteristic of cerebellum. That should be your number 1 clue that you're looking at cerebellum. So not only is it just 3 layers, but you have that real thick, distinct line of cells coming through here, Purkinje cells. And then that's your 3rd layer right there. Now I could name them, but unless you want me to, I won't. Who wants me to?
Do the Purkinje cells are just on the ground?
This is the Purkinje layer. Now that, I want you to know. The molecular and granular layer, I don't want you to know. I don't care. I said it, but I didn't say know it. No Purkinje. K-N-O-W. You need to know Purkinje. Okay, and then the last thing is the white matter. I need you to know the name of the white matter here. It's called arbor vitae. Somebody thought that it resembled a tree, and so they named it arbor, like Arbor Day. Vitae means life. So in Latin, you're looking at the tree of life. If we open up one of the cerebellum models, mine connects, but some of them split open, you can see the white matter inside, and it's all nice and branched. That's the arbor vitae. Okay, questions? Cerebellum's not bad. Next one, spinal cord. And we already saw this in the model, so it's easy. Right? Okay, this right here is the reason that I do too. That nice little structure coming up right through the center of the slide. That is what we call an artifact. Somebody's F up. That's why we didn't have a nice intercalated disc slide on your exam. Because somebody made an artifact on it and cracked the cover slip. So please be careful with the slides when you study them. And don't think that breaking a slide will make it not be on the exam. Because I will find some. And they probably are ones that you didn't study, and therefore probably won't like them. So please be careful.
Yeah.
That's just a crack, yeah. It's not an actual structure. It's somebody cracked the slide and
It's not official.
I was just trying to make a point, sorry.
You're like, what's an
Artifact, an artifact, I explained it the first week. You remember when we saw the cartilage elastic slide? There was blue and had all those crinkles in it. That whoever made the slide kind of messed up and just said, screw it, I'm just leaving it like that. Well, that's the same type of thing. Anyways, I used this slide with the crack in it because it's the only one that we have that's small. It's from a cat. So you can get most of the structures in the field of view. It's the only one that's small and has this particular stain. Much of the nervous system tissue uses this brown stain. It's called silver stain because it is so nice and selective for that particular type of tissue. Silver stain is what they used to also use for old-time photographs. Silver nitrate. That's why it has kind of that brown, old-timey look on those old photos. That's why it looks the same color pretty much as those. But anyways, spinal cord. Gray matter on the outside or inside? Gray matter inside. It's flip-flop from the brain. So gray matter, white matter. Now, please don't let the color fool you here. It is still gray matter, white matter. White matter, gray matter. This is the end of one of the horns. Is that ventral or dorsal? And you see that this is coming to the edge.
That's
This one
ventral.
isn't.
Ventral.
This is ventral. And that would be dorsal. In the ventral horn, if I zoom in, you can kind of see some motor neurons. That's all these little clear areas that don't have lines going through them. Whereas going up here, you don't really see that. These little bubble looking things there. Not that great down there. So we have ventral and dorsal. What's that? Well, it's anterior something. Anterior median fissure, posterior median sulcus, central canal. That's filled with CSF. Here we see the 3 layers of meninges. Now they are kind of peeled up. That is just an artifact. But 1, 2, 3. I like it a little bit better down here because it's stuck to the spinal cord right there for PIA. Then you have that space that's filled with CSF. That's called subarachnoid space. And then arachnoid and dura mater are stuck together up here. So pia mater peeled up, subarachnoid space filled with CSF. Arachnoid mater and dura mater is outermost. And you do have a model that shows it pretty good. I don't know where it is, but up on the cadaver tank. You can see the different layers on this 11 inches. That's the best one to sit on.
Mnemonic is that. Dura arachnoid, yeah.
Wait, what's your mnemonic for
Yeah.
it? Lateral horns, not really. It's kind of like a little bit of a bump. I wouldn't ask that. Okay. One more. This is also spinal cord. It's just a different stain. You can see the motor neurons a little better on this one. But this is from a person and not a cat, so it's a little bigger. Too much light. Huh?
Like the other one, the heart is the only one up to it. Spinal cord is the only one up to it.
Yes, it is. Okay, the main thing about this one, it's a really, I'm gonna say it, it's a shitty slide. It's blue. You have white matter on the outside, gray matter on the inside. This is a ventral horn, it doesn't come out to the edge. And you don't have a whole lot to make that discernment, but really, it's your only blue slot. You have a light area and a dark area. It's not like the intercalated disc slide where it was red and blue. It's not like your elastic cartilage slide where it was like really dark blue. The thing that I want to show you in here is your motor neurons. They stain pink. See all the pink dots that are up in there? Those are all the motor neurons in the ventral horn. So that's really what you want to find when you're studying this slide. For the other one, I'm not going to put it out because that's the only one we have. Just study your model and be comfortable with general structures of the spinal cord. Okay, we'll get through peripheral nerve and your special senses tomorrow. And then we'll have like 4 slides for Wednesday. And then I'll be it. Okay, so go ahead and study. I would get comfortable first with just like general brain structures and your cranial nerves.